Hello everyone, this is going to be the first of maybe 2 or 3 instruction videos on how to punch fin clips for this vermilion and sunset rockfish genetics project we are involved in. First of all, I thank you profusely, thank you all for volunteering to help us out. This is a real heavy lift and we couldn't do it without you. And it's not trivial, the amount of work that's entailed. It's going to be a number of hours and it's pretty tedious. But I just want to thank you all for volunteering to help out. So first video here, I'm just going to go over some of the equipment that we provided that you should have in a box that I sent to you, or if you're down in Newport, I think Aaron has a sort of a central supply repository. So just going through everything, you'll have a big stack of these plastic plates, more than enough to do however many fin clips that you've been assigned. They sort of, when they're empty like this, they're just stacked together. 96 wells, of course, per plate, and they're a little bit flimsy, so we have to be careful of that. You'll also have these plate racks, or actually I think maybe only 1 or 2 per person. So you take the plate that you're working on, and it'll fit. There's holes there. It'll fit the plate, and it just gives a little bit of sturdiness. Helps keep the plate from sliding all over. It also has a little cover, so if you're in the midst of punching and you need to take a break or something, you can cover that up. You'll have a bag or more than one bag of these little strip caps. So what these do is the orientation that we punch this, it's not left to right, top to bottom. It's actually top to bottom, left to right. So they go in this order. So your first 8 in a plate will be this first column of 8. And once you get those filled, you want to take these caps and just sort of press them down, and you'll see how they fit together. They fit in there pretty securely. They really wedge down in there, so that's good. And obviously, when you're done with the plate, all of these, there'll be a cap strip on every column when you're done. Let's go with the Finclip puncher. They're in plastic cases. I think everybody has access to at least 3 of these, and you're going to be cycling through them one at a time. So there'll be I'll talk a little bit more about that. So you should have 3 cases of these. Inside each case is 1 puncher. It's brand new, so there'll be a plastic cover on it. You can just pop that off, put it back in here, and so it's a thumb actuated deal. How am I doing this? So you can see the tip come out when you press the plunger in. That's the cutting tip there that comes out, that thumb actuated tip. Everybody also has in their case for each punch, there's an extra tip. The tip will screw off, and the idea is if that cutting tip gets dull, you can replace it with another one. That shouldn't happen given the numbers we've got here, but if you feel like it is starting to get dull and not cutting cleanly, you've got an extra tip that you can play with. Again, you should have 3 punchers. You got a cutting mat. This is just sort of a hard plastic foam mat that you're going to put under your blotter as you're punching. It just gives it so you don't damage the table, and also it provides a surface that you can clean off afterwards to avoid contamination. There's some chemicals. I provided some jugs when I didn't actually provide the chemicals itself. The first is just a 5% bleach solution. You're kind of on your own for that. Just take some household bleach and dilute it down to 5%. A jug that you can just put water. You actually don't even need a separate jug for this because we only need a little bit at a time. And then some 95% ethanol, which I believe that I did provide for everybody. I also provided a bunch of these little sort of ketchup cups that we're only going to be working with a little bit of chemicals at a time, so you can just kind of set them out on the table. And it'll be, the way we're going to work with these chemicals is bleach, water, ethanol, so in that order. And it's what it is. It's just a cleaning step for the puncher in between each punch that we do. So it's pretty tedious. Scotch tape. This is if the fin clips, the actual fin clips themselves on the blotter sheets sort of dislodge, which sometimes happen. They're very brittle. Try to keep track of If it flies across the room, try to keep track of it, and then maybe just scotch tape it back down to that blotter cell. I didn't provide these, but if you want, sometimes it can improve your grip to use like nitrile or latex gloves or even an ace bandage type of a thing. This little ring can sort of dig into you a little bit. So gloves or something like that can, and I'm not even talking about just wearing them. You can wear them, of course, but just wrapping them around the puncher can be helpful. Plenty of paper towels. You're kind of on your own for that. We're going to go through a lot of paper towels with all the cleaning and drying steps. I provided some wax paper as well. Some of the blotter sheets that people receive, depending on the vessel and year, are wrapped in wax paper that might not be in very good shape. So if the wax paper is really deteriorated, you can use these little sort of sandwich style pieces of wax paper as a replacement. Then we got you should have a manila folder of plate diagrams. And so this is the roadmap for how you do the punching. So each one of these is a diagram. So each one of these diagrams corresponds to a plate. This tells you exactly the specimen number that goes in a particular cell. So at the end of each plate, your plate should look exactly like this in terms of the specimens that are in there. It takes a lot of keeping track. I'm going to go over the specimen numbers here in a little bit. But you will see that in many cases, things may not be particularly sequential. So you can see in that first, going from that first position down one, actually goes from 1 to 3 and then 4, 5, 6, 7. So I'll show you why that's going on. You have to pay a lot of attention here and not get on autopilot. You have to really make sure what specimen number is being called for to go into a particular well of a particular plate. And I would call your attention to the plate diagram number, the plate number. It's on the bottom right, or excuse me, bottom left of each of these individual plate diagrams. So this first one is HL 169, and then the second one is HL 170. And that just means hook and line number 169 in the grand sequence of things. So one of the first things you do when you begin punching a new plate is you're going to want to transfer that number with a ultrafine Sharpie onto the two places. On the right side of the plate, you'll take that HL 169 or whatever your plate number is and write it there. And then also at the bottom, I'm not sure how well this will hold up with smearing. So the more places you can write it, the better, because if we lose track of this, we have absolutely no way of knowing what specimens are in here, because it's just a little piece of fin at the bottom of these wells, and there's obviously no specimen number that goes along with the tissue. The specimen numbers are all on the plate diagram. So the specimen number has a little bit of a code to it. So you can see on this diagram, it starts H17MI-V0001. And so and then it goes to 3 and 4. And I'll show you why it skips that, why number 2 is skipped on that one. But the code there for the specimen number is this. So the the first letter tells you what survey the clip was taken from, and I think everybody has only hook and line. I think I'm doing all the trawl survey ones. Then it's a 2-digit year number, could be anywhere from 04 out to 19 for 2019, then a 2-digit letter corresponding to the vessel. So it'll be either AG, MI, or Toronado, Aggressor, Mirage, Toronado for the 3 boats that we use. The Toronado only started working with us in 2013. So if you're 2012 or earlier, you're only going to have Aggressor or Mirage. And then you actually have the blotter cell number, so like V0361. So let me get the blotter cells out. That's the last piece of the equipment here. So you'll also have another big manila folder full of these blotter sheets. So these are the ones that are going to go out to Ken Grimes, and it's for 2017 hook and line. So it's all 3 boats. It's all the fin clips we took in 2017, all the Vermilion fin clips. And they're sort of, so you can see it's a pretty big stack. and you'll be able to see this is actually in a plate, plastic plate sheet, and it's two blotters that are stapled together. You're probably going to want to, when you actually start punching, remove these staples and put the blotter individually on the cutting mat over here. Do your punching, and then when you're done with that blotter, return it and restaple it back to that piece of paper. I think it'll just be easier for you to work with. That way you only have one blotter out at a time. But you can see the blotter sheet tells you all the information you need to know where you are. So hook and line survey, the year, what vessel, and then species. And all of these are going to be vermilion, of course. So again, it's a top to bottom, left to right. So the numbers go 1, 2, 3, 4, 5, 6, 7, 8, then starting 9, 10. So that corresponds with the plate diagram, sort of the Rosetta Stone here that tells you where to go and what to put in each plate. So you see that there's an X. So in the field, somebody marked an X. So they did not take a fin clip from that fish for whatever, the fish that would have gone there. So on the plate diagram, you'll see that 2 is missing. It goes from V001 to V003. So that's important to note. So you can't expect, and you should not expect the plate diagram. You should not expect the plate diagram to follow exactly any missing cell number. So these things will soon kind of get off synchronicity, I guess you would say. So just keep in mind what the plate diagram tells you to do is what goes in that corresponding plate. So you just have to make sure you know, oh, it skipped number 2, V0002. So you need to skip that in the well, or you need to go directly to the next specified number, which is going to be 3. I know this is confusing, so obviously it goes without saying, fire off any questions to me. You'll also see in later years of the survey, I think from 2009 or 10 and later, you'll see these 3 X's at the end. And so just follow along what's actually happening here, because there's a number 2 that's missing. Normally there are 96 cells here, but because there are these 3 X's, there's actually only 93, a maximum of 93 blotters per page. So these are always going to be skipped. Just follow along there. It may not make sense. For example, so this one ends up, this very first plate ends up 90, 91, 92, 93, 94. Well, that 94, and then it goes to those 3 X's. That V0094 is actually on not that first blotter. It goes down here to that second blotter, 94. So again, the blotter sheets and the plates are not going to be in total synchronicity in terms of the position that everything goes in. But basically, the plate diagram rules. So what it says goes in a cell is what goes in a cell. For those X's on the page and on the plate diagram, you're just skipping those. You're leaving those last 3 wells on the plate empty, and so you just follow along. And so you end that one with 94. It's not focusing. You end that plate at 94. The last 3 wells of this plate will be empty, and then you'll just move to the next plate. Your plate, whatever your new plate number is. Here it's hook and line or HL 170, and you start with number 95. You've already done 94, as I mentioned previously. I know this is getting really confusing, so I'll probably move off of this issue. Just ask me any questions if you're wondering what specimen to put in what well. It might be self-explanatory, but it might not. Let's see here. The blotters are pretty fragile, so handle them with care. Um, be very careful with the chemicals. The reason why we're using bleach is because bleach dissolves DNA, so that's a good thing between if you're trying to get remnant DNA off a puncher. It would be very bad if this bleach came in contact with this. It could denature all of that DNA there, so that would be just something to be mindful of, be careful of. Um, let's see. The blotters are pretty fragile. The fin clips themselves are very fragile. They can blow apart if you aren't careful when you punch them. If you do happen to dislodge one, that's what the Scotch tape is for. Try to find that clip and tape it back on there. The clips, as you will notice, as soon as you take them out of whatever they're wrapped in, they don't smell good. They pretty much stink. So it could make your house or wherever you're working stink up. And I'm sorry about that. It also might attract pets like dogs and cats that might want to mess with this a little bit or eat it, and that also would be very bad. So please be aware that this could attract pets. Goes without saying, you want to have a good, clean work area to keep any potential for contamination down to zero. Let's see. Sort of alluded to this. So this is the Vermilion fin clips for the Mirage for 2017, and it starts at #1 and it goes to however long it goes. It might go to 300, 400, 500, 600, depends on how many Vermilion we caught on that particular boat. But when we get to the next boat, once you're done with the boat and you're going to the next boat, say the Aggressor, that number starts at 1 again. So you have to kind of be mindful of that. To uniquely identify a fin clip, you need to know, for the hook and line survey, you need to know the year, the vessel, and then the specimen number. Otherwise, it can be ambiguous. In 2017, there were 3 fin clips with the number 1, or V0001, as we call it in the database, and on your specimen number. So you need that. You need that middle. You need to know what boat is also going along with that. So this is all of the Vermilion fin clips for all 3 boats for the hook and line survey in 2017. Let's see what else here. Keep a running log of any questions you have. You can email me right away, but if there's any irregularities that you see, like, wow, why is the plate diagram skipping this number when it doesn't look like it should? Why is there something that's not following this pattern? If you see something in there, there are going to be some occasions where these are totally out of sequence. It'll skip, 5, 10, 20 fin clips, and there's usually, it's either a mistake or there's a reason for it. And make a note of that, and if it doesn't make sense to you, let me know, and I'll look up why that is. There may also be some errors in here with the macro that I used to put this together. And that could be just a Word document or just an Excel spreadsheet that you keep of notes to yourself. Okay, that's it for this video. We're going to go to one that's going to kind of show you what what your workspace might look like. Okay, rather than doing a totally separate video for sort of your workspace, just paused it there. So this is what it might look like. You've got a kind of a nice table layout that you've cleaned off, free of contaminants. I have separated that double blotter sheet. I took the staples out, removed that top blotter, put it on the cutting mat here, and I paper clipped it so it doesn't move around, because you are going to be pressing the puncher into that, and sort of there's a potential for leaving residue behind. So you don't want the blotter sheet moving around a whole lot. It's not that big of a deal, but if you can avoid it, you don't want the blotter sheet moving around on the cutting mat if you can help it. because you might pick up residue from a previous punched clip. And you will be, in between each blotter sheet, you will be wiping before putting a new blotter sheet down. You will wipe it down at the very least with a little water and a little ethanol. You can bleach is good too, that 5% bleach solution first, but you definitely want to make sure that you rinse all of that bleach off, and that you finalize, you put a little, you finally wipe it down with the 95% ethanol. So here's your blotter. Here's your the active plate that you're going to be working with in your rack. In the previous section clip, I already put the plate number on it, which corresponds to your plate diagram. So you'll have your plate diagrams. This is kind of how I have my set up. You got your 2-3 punches, probably 3 that you're going to be working with. Here I just have a few cap strips out to cap as I go. I got my bag here to grab more as necessary. Up here, got the chemicals just to show you what's there. It's left to right. You've got the 5% bleach, just regular tap water, clean tap water, and then 95% ethanol. Of course, for the purposes of this video, it's just water in all 3 of them. And what I'm learning is these ketchup cups may not be the way to go, because I don't think that they're going to hold up with liquid in there for more than maybe an hour or so before they start dripping out. So what you can do is you can actually just use the caps of the, or maybe even better, get yourself just a cap or something, a clean cap about this size, because all you're going to be doing, all you're using it for is you're going to be, it's a cleaning process. So after each punch, you're going to dip it first in the bleach. You're going to want separate paper towels for each solution. So you first start by the bleach, rinsing it out real good, then going into the water, activating the punch a few times, drying off the water, pressing the water. You want to get pretty dry, and then going into the ethanol. And the reason why you want to dry a little bit between each one is you just don't want you don't want any bleach going into the water, or not that much. So you want this to be a pretty clean solution. You don't want to be carrying, once you're done removing any remnant DNA with the bleach, you don't want to retain that bleach to go into the water and then into finally the ethanol. And then you'll do a final rinse in the ethanol, and again, you really want it to dry out. I even have a final one here, and then what you do actually, as you're ready to move to the next punch, is just set that aside. Just set your puncher aside, and then move to the second one and punch your Finclip, and then put it in the plate, and then do your rinse, and you set that one aside, and then you do the third one. And the idea is, and then you just cycle through them 1 through 3, and you go through them one at a time. And the idea is you're giving yourself a few more minutes to let these dry, because if any water remains on that tip, it makes it really tough. It will suck the puncher up in there, and it won't come off cleanly. It'll suck the Finclip punch up in there, and it won't come off cleanly when you try to punch it. So you want these to be as dry as possible in between. As you go, I'm not going to show the Finclip punching technique just yet. I'm going to have Victor come out and hold the camera for me. But again, so this is just sort of the workspace that you've got lined up. You're going to be using your plate diagram as the guide. It's saying HL17 Mirage V001. We already know from this what that corresponds to. You should have the appropriate blotter in front of you. You'll punch that. You'll punch that clip. You'll put it in that well. You'll do your cleaning step. You'll move on to the next, and it says V003. Of course, 2 we saw before was skipped, so you go straight to 3. You punch that, and that'll go into that very next well, exactly as how it's specified on the plate diagram. And then you just move your way through it. And then once you've done 8 at a time, once you've filled that column 1, 2, 3, 4, 5, 6, 7, 8, you put that cap strip on. So you'll press that down in there, and then you just keep on moving So now you're on this second column, which H17 Mirage V0 0010. So that's Mirage 2017 number 10, and you just keep working your way through it. And then, okay, so I'll pause here and then we'll go into actual Finclip punching technique here. Okay, so now this is probably going to be the final piece of this video. I've got my assistant Victor here. We're going to go over actual punching technique. So I've gotten my 3 plungers. These are brand new, never been used before. So just prior to this, I actually did the cleaning step with the bleach, the water, and the ethanol. And I got rid of those ketchup containers for the bleach and the ethanol, and I'm just using, I found some plastic caps. I just think these ketchup things are gonna just get saturated, in fact, even with the water ones. So I would recommend just finding even some bottle, like some like a Gatorade cap, or even like a soda bottle plastic cap that you can clean out and use, because you don't really need a whole ton of the liquid. But those ketchup containers, ketchup cups, really aren't going to work the way I thought. So, okay, so we're going to go through technique. Just a quick review. I've got my plate, my active plate that I'm working on. It's in the plate rack, and you can see it's already been labeled HL169. And over here to my left, I have my plate diagram, and that HL169 comes from here. Again, each one of these is a brand new plate that you'll start, HL169. And now I have my blotter here. I've got my punch, and so we're going to go through the technique. You see I've got it paper clipped down to the cutting mat to keep it from moving around. So you only do 1 punch per well, and then after the cleaning step, you move to the next. So you don't need to do 2 punches, and to get more tissue in there. 1 punch is plenty, and it doesn't really matter too much where on a choosing where to punch the blotter itself. The diameter of that actuated tip is maybe a couple of millimeters or so, so it'll take a pretty good sized punch out of it. I try to go where it looks like there's a fair amount of tissue, and I try to avoid areas that are really built up, like with a lot of fin ray. If a fin ray is stacked on itself, that could potentially damage the fin clip on the blotter and cause it to come off. So for this one, I'll just take it right here. And so the technique is you just kind of hold it in your fist, and then you press it down and you apply some pressure. And I go in like a circle pattern there just to apply some pressure. And as you can see, it'll actually cut through the paper, and that's totally fine, not a problem. Actually, this is the diameter of this punch is actually larger than the one that I'm used to. But again, so I have punched the first cell on that blotter. I know that over here on the plate diagram, it's H17 Mirage V001. I just verify. The plate diagram tells me it's going to go into this first position, so the A1 position. Basically, you're just following the orientation of the plate diagram here with the plate. So all you do is you put the tip in that well, kind of, you can get, I don't know if you can see in there, Victor, kind of maybe going from the side, but basically you get the plate actually into the well, and then you activate the plunger, and you can see it came out. So that's great, perfect, very nice. So that one's done. Now what we do is we do the cleaning step, and it's a 3-step process I talked about. It's to remove contamination, and then also make sure all the remnants of bleach are off of it for the next one. So you go into the little bleach here. You just have to dip in the tip a little bit, activate it a few times, kind of pressing that down. And then go over here to the bleach paper towel. I use the separate paper towels. You're probably going to have to go through a bunch of these each Finclip session. Get the majority of the bleach off, then go into the water, do the same thing. Try to show there, just sort of activating the plunger in the water, tapping that off, getting it as dry as possible, then going in the ethanol. You're just trying to not cross-contaminate the chemicals very much if you can avoid it. So this ethanol is the last step, and ethanol dries pretty quick. It's really good at getting the last remnants of the ethanol. It sort of deplaces, excuse me, the water, deplaces the water. So you let it dry, and then I even have this final little drying area. And so, like I said, we're going to rotate through these. So we have 3 to work with. So we're going to put that over here, and then move to our next one. And that'll just give that a little extra time to dry because you don't want that tip to be wet at all. I've already pre-cleaned all of these. They're brand new. So we go to the next one. If you remember on this is kind of a unique, this is not necessarily how it goes. Number 2 is missing, so it goes from 1 to 3. The reason why it's missing is because in the field it was X'd out. So there's nothing to punch there. We won't punch it. We're going to go straight to the next one. In the plate and the plate diagram, we're not trying to match everything. So there's not a missing cell for that particular X that's marked in the field. So we're going to go straight to number 3. Find a good place to punch it. This seems like a good place. You can hear it sort of crunching there because it's crispy and whatnot. You got to be careful. Okay, got a punch. I'm going to go over here. Again, it's going in the second well of that first column. V003 is going in that second well. Again, activate the plunger. You can see it popped out. Some paper came with it. Totally fine. No worries. Again, now it's the cleaning step. Drying. Water. Drying. Ethanol. Drying. Some more. And I'm pressing the plunger down to try and shake some of that water off. Again, that's next in the rotation. Now I'm going to look at the plate diagram. It says to go to H17 Mirage V004. That's number 4 there. So boom, we're going to do it. We're going to punch it right here. Get it, kind of rotate a little bit, a rotating action. Not a rotate, but sort of rock it back and forth gently, gradually increasing pressure. And you want to make sure that you have gone through the paper. Most times it will go through the paper, and that's a good indication that you've got the clip there. So we go into the third well, which is where the plate diagram says to put it. Flip it in there. Go back to your cleaning step, bleach.
Have you had an instance when the paper plug did not come out of the plunger?
That does happen, and these ones are brand new. It happens sometimes when a cut is not that clean and there's edges to it. These are all brand new punchers and brand new cutting tips, so that's probably not going to happen much, if at all. It also happens when you leave too much water. If you were to go back in and try to punch that again, there's still some water residue, or ethanol residue, I should say. That will kind of gum up the works, and you won't necessarily be able to punch it out the way you should. But that's why we have this sort of 3 punch rotation to give extra time. So I'm going to finish up this column off camera, and then we'll come back in what you do when you're moving to the next column. Okay, so I've gone ahead and punched the first 8 Finclips here on the plate diagram. So I'm done with this column. You can see that there are clips in each one of these wells. So a very good best practice is, as you're done with the column, cap it right away, because it can get very tedious trying to match up exactly where you are on the plate and plate diagram. So just to give yourself a little kind of a baseline to work from, cap the that column, each column as you go, so then you know you're always on the one, the first, you're in the column, the first open column. So that should wedge in there, and so you'll just work your way down. If you need to take a break or something, you can kind of cap this up, but you'll work your way through until this is done and everything is capped off. When you're done with plates, put them somewhere. It might be a good idea to possibly put each plate in like a small Ziploc sandwich bag, because they are pretty flimsy. I'm worried that the caps could work themselves out maybe. So I'm thinking maybe a Ziploc sandwich bag that you can seal up, and then maybe put a couple wraps of Scotch tape or something just to keep the bag tight against the plate. You just don't want the caps popping off. Let's see here. Just a couple of things. Not sure if just to emphasize, the bleach solution is actually a 5% bleach solution. So, you take your household bleach and you dilute it down 5%. When you're done with each blotter cell, and remember, the plate diagram and the plate match each other 1-to-1 in terms of position. You may get a little bit off, and you will get a little bit off. They won't always correspond in orientation. So, for example, because this, let me show you a picture here. Because no fin clip was taken from that fish in the field, in order to get 8 punches, I actually had to go to the first row of the second column to finish that first. So you will be off. Your plates and plate diagrams won't be off; they'll match 1-to-1, but you'll be a little bit off. But when you're done with punching all the fin clips that are on a blotter sheet, you'll take it off. It's a good thing to maybe have a paper towel that you can put a little bit of that 5% bleach solution, just a little bit. You want to get any maybe remnant DNA that might be there. Let that dry a little bit, and then maybe do with a separate paper towel a little water wipe down and then a little ethanol wipe down. It just should take a couple of minutes for it to dry, but that's a good practice to do in between each blotter.
Just a quick question about your bleach solution.
Yes.
When you say 5% dilute, are you diluting it with just regular tap water?
Yeah, that's correct. Absolutely, just 5%. So you'll go and you'll get your next blotter sheet. You can staple this back onto the page, pull the staples back up off of this one. You'll move on to the next one, paper clip it down. In some earlier years, they're not stored this way. They're stored in pieces in wax paper. So just put them back in the wax. Basically, put the blotter sheets back the way they came, the way they arrived to you. If it's because this can be a really tedious thing trying to match up this plate diagram. It's small numbering with these little wells. It is certainly possible you may get off. You may put a punch in the wrong well, and I need to actually find out what to do about that. I think you can probably just remove it and kind of get the remnant out as much as you can. What you could do is if you're starting to go down here and you put the wrong plate, maybe you could sort of do a partial cap so that what's in there stays in there, and you just want to remove what's in there. You could flip it over and maybe wipe it out with a little bit of ethanol inside there, maybe with just the corner of a paper towel or something. Obviously, do what you can to make sure that you're following along correctly, so that doesn't happen. Again, make notes of any irregularities you see. If there's if there's an unexplained skip in the plate diagram, like, oh, this goes straight from 35 to 37, but it's not X'd out on the blotter sheet, what's going on? Make any note of that and just try to move on, make a note of that and just try to continue on matching what the plate diagram does say, and I'll try and figure it out after the fact. Let's see here. When you're at the end of, usually it's when you're at the end of a year vessel combination. So when you're done with all of the Mirages, so there's probably, I don't know, probably 7 or 8 blotter sheets just for the Mirage 2017. And that, the last blotter sheet for a year will end somewhere in the middle. And what I've done there, that's all we got. So what I've done there is your plate diagram will end there as well. So there'll be the last several will be of every year vessel combination will be empty. And that's fine. You just need to make sure that all of the columns that do have Finclip punches in them are capped. You certainly don't need to cap columns that don't have any specimens in there. Again, I can't emphasize enough, please do keep a running log of any missing or bad, what looks like bad sequences. You might even note if a fin clip looks particularly terrible, or if a fin clip is missing. But if it's really moldy or something, maybe make a note of that because we want to see, um, we want to get an idea of what the shelf life of the DNA is on some of these that get moldy. Um, there's going to be a small number where there'll be a small number, there's none on this set, but where the cell is highlighted, like I shaded the cell, and that usually just means to, pay close attention because I've tried to identify things that are out of sequence. Um, so just follow along with what's in the diagram, even if it goes straight from 127 to, if that went to 135 or something, you would put 135 in there, but just make a note, hey, 127 to 135, there was a gap, and I'll take a look at it. Let's see. And then once you have about 10 or so plates that are full, we can start talking about how to get them back. So if you're really burning through this and you've got 18, 20 plates that are assigned to you, and it's not taking you that long, you can just wait and finish them up, and we'll figure out how to hand these off. We're probably not going to be shipping them because we don't want to lose these. We'll probably organize some sort of a meetup somewhere. If you're in Newport or obviously in Seattle, you can just bring it by my office. But if it's taking you a while, like several weeks, and you get about 10 done, well, let's figure out a way to hand them off, because we want to send these off to the vendor who's doing the genotyping, the actual genetics PCR work. We want to keep him busy and keep giving him product to work with. But we'll figure that out. I think that's about it. We'll probably have a little, I'll probably schedule maybe a Google Hangout for all of us after you've had a chance to receive your supplies and review this unexpectedly long 42-plus-minute video. Sorry about that. We'll probably do a meetup or a hangout or something like that to go over any questions you have before you get started. If you feel like you got it all nailed down, I guess go ahead and get started. I think we'll still probably do a little meetup just in case there's some questions and to get on the same page. So, and if at any point during this video you had some questions, hopefully you wrote them down. Just either call me or email me. And I think that's it. Do you have any questions, Victor, that might be helpful? Okay. All right. Thank you very much. And once again, I do really appreciate everybody's help with this.